Alzheimer's disease, the dysfunction of protein folding and inhibition of acetylcholinesterase, presented by Abimbola, Johnson, Olabode, and Courtney Campbell. The article we have chosen to base our presentation upon was found on the PubMed website, authored by Courtney et al. In the year 2008. The name of the title is Effects of Familial Alzheimer's Disease, Mutations on the Folding Nucleation of the Amyloid Beta Protein. This article states the self-assembly of the amyloid beta protein, abbreviated AB, into aggregates ranging from small oligomers to amyloid fibrils, linked causally with Alzheimer's disease, abbreviated AD. The image shown on the presentation slide is a diagrammatic structure of several mutations of the amyloid beta protein. To familiarize you with the abbreviations, WT stands for wild type, E22G represents Arctic, E22Q represents Dutch, E22K represents Italian, and D23N represents Iowa. C1 and C2 represent cluster 1 and cluster 2.
Here you can see an example of how a peptide bond is formed. This is a condensation reaction, and you can see that a molecule of water is eliminated from the reaction. This allows the alpha carbon atom and the amino group to link together, which forms the peptide bond, which is shown in the diagram. The rigid peptide group that is adjacent to the pure single bonds, which are between the amino group and the alpha carbon atom, which is circled in green, and the alpha carbon atom and the carbonyl groups, which is circled in red. These are able to rotate, which gives different orientations to the primary structure. Because the two bonds of each amino acid can rotate, it gives the ability for the proteins to fold.
The folded polypeptide chain gives several identified secondary structures, which are the beta turn and the omega loop. The alpha helix and the beta-beta sheets. These structures are formed by a regular pattern of hydrogen bonds between the peptide NH, nitrogen-hydrogen, and C-double-bond-O, carbon-oxygen groups of amino acids that are near each other in a linear sequence as demonstrated in the alpha helix and the beta-beta sheets diagram. Together they form a protein's final tertiary structure.
As you can see from the given diagram, tertiary structures tend to be water-soluble with non-polar cores. This is because, due to being in an aqueous cell environment, the protein folds in a way that the hydrophobic residues are excluded from the aqueous environment and the polar charged chains are on the surface, hence why it has a water-soluble structure.
Newly synthesized proteins need to be folded correctly and amino acids need to be chemically modified after protein synthesis. Once a protein is folded correctly, it is in its native conformation. Misfolded proteins are referred to as non-native. The amino acid sequence of a protein determines its three-dimensional structure. However, the environment of a cell is aqueous. This poses as a problem because an aqueous environment does not favor the correct folding of the majority of proteins. Therefore, proteins are folded correctly with the help of chaperones. Chaperones are a class of proteins found in all organisms. A group of chaperones, called the Grote chaperonins, form a large multi-subunit complexes which are called chaperonin folding machines. It is suggested that newly synthesized proteins enter a chamber of the chaperonin folding machine, and this machine creates an electronically neutral microenvironment so that the newly synthesized proteins can fold successfully into its native conformation.
Protein misfolding can lead to diseases such as those stated on the slide: mad cow disease and Creutzfeldt-Jakob disease, which is also known as CJD. The diseases are caused by agents called prion proteins, in short, PRP proteins, and the diseases are transmitted by aggregated forms of these PRP proteins. The protein PRP is normally found in the brain, and the main bulk of its structure is extensive regions of alpha helices and very little beta strands, as you can see in the diagram on the left-hand side on the slide. The aggregated form of this protein is called PRPSC, which you can see on the right-hand side diagram. The protein aggregates have a range of molecular weights and are resistant to treatments that are usually used to degrade most proteins. And because these protein aggregates are insoluble and have a heterogeneous nature, the PRPSC structure has not yet been determined. However, as you can see from the diagram, evidence suggests that the alpha helical parts of the PRP protein are converted into beta strand conformations. This leads to the beta strands of one protein linking with another beta strand on another protein. This will form beta sheets that join the two proteins together, therefore leading to the formation of protein aggregates. The fibrous protein aggregates are called amyloid forms. The protein aggregates that are built from the abnormal PRP protein act as nuclei which other normal PRP proteins attach to. Therefore, the prion disease can be transferred from one organism to another through the transfer of an aggregated nucleus. This can be seen in the formula that is below the diagram. Non-infectious neurodegenerative diseases like Parkinson's disease and Alzheimer's disease involve amyloid fibers. In this presentation, we'll be using Alzheimer's disease as the example.
In patients with Alzheimer's disease, their brains contain protein aggregates that are called amyloid plaques. These amyloid plaques consist of a single polypeptide called amyloid beta, which is derived from the amyloid precursor protein referred to as APP via the action of specific proteases. The polypeptide amyloid beta forms insoluble aggregates, so having to use a nuclear magnetic resonance spectrum, NMR spectrum. It was found that amyloid beta protein structure was rich in beta strands, which form extended parallel beta sheet structures.
A form of treatment for Alzheimer's disease is the tablet donepezil, what is most commonly known as Aricept. Aricept is a competitive acetylcholinesterase inhibitor. Because it is a competitive inhibitor, its effects are reversible and not permanent. Therefore, taking the drug is an ongoing treatment. Normally in the brain, acetylcholine helps messages travel from one end of a nerve ending to another. After that, an enzyme called acetylcholinesterase reduces the amount of acetylcholine being sent through. Acetylcholine is what helps maintain brain function with things like memory, awareness, and judgment. When someone has Alzheimer's disease, they have less acetylcholine, which is what causes the patient's symptoms.
Aricept helps by increasing the acetylcholine by slowing the effect of acetylcholinesterase by competitive inhibition. As the disease progresses, the acetylcholine levels decreases further, and so higher doses of Aricept are given to try and increase the amount of acetylcholine. Unfortunately, it is not a cure for the disease, it only aids in treating symptoms for the disease. The dosage is usually started at 5 mg per day, and after 4 to 6 weeks can be increased to 10 mg per day, and then further after 3 months or more, it can be increased further 23 mg per day. Thank you for listening to the presentation.